Hi everyone, again, I'm Kristen. I'm super excited to be here, and I'm going to talk to you today about linking genetic variation to nutrition phenotypes, specifically using genome-wide association studies. And I know association may seem like a very simple term, or alternatively you may be thinking, this seems like way too much math, I cannot learn this much statistics. Whichever spectrum that you are on, I really hope that you will ask me questions. You would actually be doing me a huge favor. I am a student like you, and I am early on in the process of my research phase for my PhD, and getting questions from all of you will really help me solidify my own understanding. So please speak up. This is supposed to be sort of fun and to give you a really intuitive understanding of what we're trying to do with genome-wide association studies. In what context do we use them, and in what context are they not really appropriate? So I'm actually going to spend a significant portion of this lecture just going over the mechanics of what a genome-wide association study does, and then near the end we're going to why don't I just move to my next slide, which is going to sort of tell you what we're going to do today. So the first thing we're going to do is I'm going to explain when researchers use genome-wide association tests to study a trait or disease. And then we're going to do a quick activity looking at the paper that hopefully it looks like a lot of you read, awesome, and answer the questions on Sakai. And then we're going to go through some limitations and sources of bias that come up when conducting genome-wide association studies, and how these sort of impede clinical application or further study in the lab to understand the biological mechanisms underlying disease and traits. And then lastly, I'm going to describe for you a few more real-world examples of genome-wide association studies using nutrition-related traits and diseases, and go through with you my current proposal, which is still in its early stages. Any questions so far? Awesome. Okay, so why study genetic variation? So the main contribution to common traits and diseases is often through the genetic component. However, these traits and diseases have no single cause. And so I want to lay out one of the very important definitions, which is just that multifactorial traits. And these traits are controlled by a combination of multiple genetic and environmental factors. So I know this may seem really obvious, but I'm just going to go through a simple schematic that I hope can really solidify this for all of you. So for instance, we have a population with any disease, which for today I'm calling disease X. And then within a population, you might get, within this larger population, everyone of which has disease X, you might have some subpopulations, and these subpopulations you can't tell from their phenotype, from the trait or disease that they have, that they're part of these subpopulations, but in fact their disease is caused by slight differences in the biology and the genes underlying the disease, as well as what environmental exposures each of these subpopulations are exposed to. So we know that the first group of genes and the second group of genes and certain environmental exposures are all increase susceptibility to disease X. But we don't actually know ahead of time which patients belong to which subpopulation. So how can we identify which genes are the most important in most patients? And the key assumption that we make here in genome-wide association studies is that important genes that increase the susceptibility to disease X are present on chromosome segments that are shared by many patients in the study group. And I hope that it's sort of makes sense to you. The corollary to that would be that most gene variants that increase susceptibility to disease X are found on these shared ancestral chromosome segments. And that's an assumption that we make, and we'll kind of go through as we progress through this presentation, like when that would not be appropriate, when that assumption does not hold. Okay. first, what do I mean when I say shared ancestral chromosome segment? So in this figure, this is the top of the evolutionary tree where each branch point over time represents a generation in the population. At some point in recent history, a mutation occurs at a particular chromosome segment in one person in the population that increases susceptibility to the disease X. And that's indicated just by the little red star there. And then a number of additional generations pass until we get to what we're going to call present day in this example. And we have a number of people here on the very bottom of the population tree here that have disease X or are not, represented by the blue and the red dots. Now you can see everyone in the shaded region at the bottom has descended from the common ancestor who had a mutation increasing the susceptibility to disease X. That's why there's a greater proportion of people in the shaded oval with disease X, so 7 out of 10 people, I think I did my math right, or 70%, compared to the proportion of people with disease X outside of the circle, and I think that's, I added it up, it's 7 out of 22 or so, which is about 32%. So you can kind of see, this is me just tabulating it for you, but you can essentially say, okay, who has disease X? 7 out of a total of 10 have the ancestral mutation versus 7 out of 22 don't have the ancestral mutation. So you might be thinking this is all well and good, but how do we actually know what these risky ancestral mutations actually are, right? We can't go back in time and sequence the variants in everyone's grandparents and great-grandparents and great-great-grandparents, etc., back hundreds of generations. That's not feasible. So the straightforward answer is that we don't know 100% what those ancestral chromosome segments actually are, but we can make a pretty good guess in which we look at all the ancestral chromosome segments across the genome in our study population based on what we know about how chromosomes recombine during meiosis and some hopefully very simple math. Okay. So for instance, let's say that we have this study population in yellow, including a mix of patients with both disease X and controls. So for this example, we have exactly 12 people in our study, but I hope you can all appreciate that in genome-wide association studies, we usually have considerably more than 12. But I think this will illustrate my point. So then we collect a bunch of data on 1 million or more genetic variants spread across the genome for all 12 patients in our study population. And these 3 consecutive genome positions happen to be 3 of those millions of genetic variants that we actually measured. Then we identify possible variants at these 3 consecutive chromosome positions. And I want to make it clear for this example, I'm only showing 1 chromosome, but obviously all of us are diploid. So in a true study, we would be looking at both chromosomes, but for the same example, we're just going to look at 1. Okay, so in this example, each patient in our study population has either a C or a T at position 1. They have either a C or a G at position 2, and everybody has a G at position 3. Got it. Okay, so then we determine what proportion of patients in this population have each of these possible genetic variants. So for instance, how many people have a T at position 1 versus how many people have a C. And in this example, we've said it's about a 50-50 split. And then the same thing for position 2, we have a 50-50 split between C and G, and at position 3, everybody has G. Okay, so now that we know all the possible variants at our 3 positions and the frequency of each of those variants in our population, how do we determine whether any portion of this chromosome is, quote, ancestral? To call a chromosome segment ancestral, the consecutive positions in the segment must be inherited together, right? That's sort of the underlying assumption of when chromosomes recombine are positions that are closer to each other, inherited together. Said another way, in sort of more like math lingo, so positions 1 and 2 are considered ancestral. If given position 1, you can always predict position 2, or vice versa. If you're given position 2, can you always predict what will be in position 1? Okay, so to identify the ancestral segments among positions 1, 2, and 3 in our study population, we first have to establish the combination of variants we would expect to see if each position was inherited completely independently. So let's figure out what that combination frequencies we should expect to see if all 3 of these positions were inherited independently, given the frequency of the variants in our population. Okay, so hopefully it's fairly obvious to you that if each of these 2 positions were inherited independently, we would get 3 people with a C at position 1 and a C at position 2, 3 people with a C at position 1 and a G at position 2, and again, so forth, 3 people with TC and 3 people with TG. And if you're math inclined, that's just a joint probability. But it should be very obvious from the picture. Okay, and then let's do positions 2 and 3. We can repeat the same process to figure out what combination of frequencies we should expect to see if positions 2 and 3 are inherited independently, given that we have a 50-50 chance at position 2 and a 100% chance of G at position 3. And again, this time we would expect 6 out of 12 patients to have each of the 2 variant combinations, and again, just simple math. So this is what we would expect to see. So what do we actually observe? So now that we know what variants we should expect to see and in what proportions at these 3 positions in our study population, assuming that all variants are inherited independently, do we actually see these proportions in our study population? Okay, so what we actually find in our study population, which is this time in the light yellow square, is we see 6 patients with a T and a G, and 6 patients with C, C, and G. And what can we say about this? So, kind of like I alluded to earlier, the variant at position 1 always determines the variant at position 2 and vice versa. But variant at position 3 does not determine the variant at position 2. So we would say that position 1 and 2 are associated with each other. So this is an association that has nothing to do with the disease. This is just an association with the positions in the genome. While positions 2 and 3 are not associated. And for some statistical genetics lingo, this is called when positions 1 and 2 are considered to be in what's called linkage disequilibrium. It's probably a better term would have been allelic association, but this was the term that was given and it has stuck. And it sounds very fancy, but that is literally all that it is, just an association. And for some more lingo, which perhaps you've already heard, so in this example, T and G would be considered a haplotype, and C and C would be considered another haplotype at these 2 positions. Okay, so we assume that this set of haplotypes across the genome for our study population comprises the ancestral chromosome segments. Good? Awesome. Okay, so once we have this set, we can simply go back and ask, what ancestral chromosome segments across the entire genome are associated with disease X? Okay, so hopefully any questions before I move on? Okay, so hopefully maybe somebody in the room was kind of like, what is this other dot over here? This other ancestral mutation that only 2 people in the population have from this figure. This other ancestral mutation also increases susceptibility to disease X. However, there's only 2 people in the study population that have this second rare ancestral mutation, and so we call it just rare mutation. And compared to the first ancestral mutation we discussed, which was found in 10 people, this one is only found in 2. Okay, so because the second mutation is so rare, when we go to perform the calculations to decide, to determine all the ancestral chromosome segments in our population, we can't actually detect that the variants in this region of the mutation are associated with one another. And I'm going to take you through that example. You don't have to believe me. So let's go back to our example. So remember when we were determining the haplotypes from our observed data, we had to figure out that position 1 and 2 are associated with each other, while positions 2 and 3 were not. And we could do that by comparing what we observed with what we expected, given the proportion of variants at each position. Well, what if only 2 of the patients, like in the figure, in the study population had a rare ancestral mutation that increased susceptibility to disease X? And let's say that that rare ancestral mutation resulted in a T at position 1 and a C at position 2. Could we actually detect that rare ancestral mutation in this different, now this is a different study population, not the same as the one we started out with. So keep in mind that since this is a new study population that includes the 2 individuals with the rare variant TC, we don't have a nice 50-50 split between the possible variants at positions 1 and 2. Instead, we have 5, hopefully you can see this, but at position 1, we have 5 patients out of 12 that have the C variant, whereas 7 out of 12 have the T, if you look at these 2, those 5, and then so forth. We have 7 out of 12 have C, these 2 groups, and then 5 out of 12 have G. And again, everyone has G for position 3. Okay. So just like for the previous study population, we first collect data on the genetic variants at these 3 consecutive positions, and which will tell us the type and frequency of these variants in our population. So all we know is that we have a population full of case controls, or people with disease X and controls, and we have our possible variants and their frequencies. And again, like last time, we determined the number of patients in the study population you'd expect to find with each possible variant combination at positions 1 and 2. And some math. But you can see it from the picture. This is what we would expect to see. But what do we actually observe? So in this case, we see that, jumping ahead, in this case, we see in our actual data that there are 5 people with the CC haplotype, there are 5 people with the TG, and 2 people with TC. So CC is a haplotype in this example because it occurs significantly more often than expected by chance. We have 5 people that we observe with C and C at positions 1 and 2 when we expected that there was only going to be 3. And then, again, TG is also called as a haplotype because it occurs more often than expected by chance. Again, 5 is greater than 3. But TC is not a haplotype because it occurs equal to or less than often than expected by chance. And so we don't, despite knowing that there are patients in the study population with the variants TC at positions 1 and 2, we can't actually call it a haplotype in our study population. And then sadly, because it's not called a haplotype, we ultimately cannot test whether this rare ancestral mutation is associated with disease X. So I hope that it's obvious to you here. The take-home point is just that we cannot test rare variants with genome-wide association studies, and hopefully this sort of makes sense as to why.
Can I ask a question?
Yes.
So I know that probably different people have different definitions, but what would you call a rare variant versus a common variant? What frequency cutoff would you use?
It really depends on, I mean, most people use like 1%. But I've seen people go as low as 0.1%. It really depends on context, and and what technology you use to interrogate each of these SNPs or variants, depending on like how robust so the data that's collected in a lot of genome-wide association studies, I'm not sure if you've covered this already, but are in SNP arrays, and they're probes that like light up and they tell you, okay, this is this variant is here because it gives off some intensity of color, and that's measured by like a microscope or a very fancy microscope. If you use a different data type and you do more like deep sequencing, and then you're interrogating a lot more positions, and you're more confident in your calls, people will sort of move that line to be, they'll have more confidence in saying like, yes, we can reproducibly detect this variant at, you know, a lower threshold. Hopefully that makes sense.
But 1% would be kind of a good rule of thumb.
Yeah. I avoided using the word SNP in this presentation, and it's only because I do think it is sort of, it was confusing to me, maybe it was very clear to all of you, at first, like to refer to like the same thing that's happening, like a real person has a variant in their has a single base pair change in their DNA, but having a term that refers to like a whole population can be sort of, I mean, I thought it was confusing at first. And so I sort of defaulted to discussing variants because I think it's at least more, I don't know, ambiguous in that it requires you to ask me and actually know what I'm talking about. In this case, I'm talking about a single base pair change. So, any more questions? Okay, so then this is the best part of the study. At the end, after you've done all of your figuring out what your ancestral chromosome segments are and doing a whole bunch of other data cleaning that is sort of the bane of every researcher's existence. We are first going to keep variants at one position per region of linkage disequilibrium, and hopefully it's obvious to you why we're doing this, right? If we know that we can always predict what is at position 2 from position 1, we don't actually need to test position 2, right? We can make the assumption that, okay, if we have a C at position 1, we're always going to have a C at position 2, and so we want to know if that block, that segment is actually associated with disease. And that's what I'm just saying with number 2, we test this representative variant for association with disease X. In this case, it'd be either C or T, and is that associated with the status of the patient's disease X or control. Okay. Yeah.
So I know you're going to get to this, but just keep in mind that we have like 3 billion base pairs, and most SNP arrays are maybe 500,000.
Yeah.
So
some are getting even more. But
yeah. So that's how you can cover the genome with a fraction of the total base pairs because of this association.
And it's sort of beyond the scope of this lecture, but perhaps some of you may have been a previous life or covered it in your training thus far. This is just a simple association. So if you're familiar with a chi-square test or a Fisher's exact test, that's what we would be using to test for this. We would just have a contingency table, a table like, do you have disease or not, and do you have C or T at this position. It's fairly straightforward statistical testing. There are obviously modifications on this, like you can take into account and if you have any, like we always usually consider people's sex and their age, because a lot of times the influence of a position on disease could be an important confounder, and so that's something else we would include in our models. And you can also take into account whether or not the effect that you would get by having a C. If you actually had 2 chromosomes, right, you can imagine, well, is this going to be an additive effect? If I have 2 Cs, do I have twice as much disease or a trait in this case? or do I have some intermediate? So basically it's modeling recessive or dominant inheritance. Okay, so hopefully I have taken you through this. Be able to explain when researchers use genome-wide tests to study a trait or disease, and that's to find common genetic variants associated with multifactorial traits. And I really like this picture. This was from a review, Nature Review in Rheumatology, and it just was a really, I thought, a very pretty picture, sort of hammering home what a linkage disequilibrium, that we have some ancient DNA sequence, and that over thousands of years, de novo mutations arise, and there's a number of recombination events with every new generation. And that results in our modern DNA sequence. And this modern DNA sequence has a number of linkage disequilibrium blocks or haplotype blocks. And within that block, we actually test various SNPs. And within this block, you can see there may be genes that are not really important in contributing to the susceptibility to your disease of interest, while there may be variants in other genes that are truly causative. Okay, and this is another definition that I was hoping all of you could take away from today, which is just that association studies can only identify factors that are present on chromosome segments that are shared by many individuals in the study group. Thus, their success depends on what is called the common disease, common variant hypothesis, which supposes that most susceptibility factors are ancient variants found on shared chromosome segments. So, basically, my hope is that you will leave here today knowing that even if that a rare variant is just not something that we can test with genome-wide association studies, that it's set up on this assumption that that common diseases have a genetic component that results from these common variants. Okay, and then we can't go any further without talking about this issue of effect size, which is the magnitude of the association between variant and the trait of the disease. Up until this point, I've just sort of said, oh, it contributes. But you can imagine if you have some sort of quantitative trait like vitamin D, that there could be an amount that each SNP is contributing to that outcome. And so when it comes to studying GWAS, we're over here usually. We're looking for common variants with low effect on complex or multifactorial traits. And that doesn't mean we couldn't identify common variants with high effect size on various complex traits and diseases. But for the most part, many of those variants, if they're very extreme, have been identified, had been identified before actually embarking on the era of GWAS, which was early 2000s, after the Human Genome Project was completed. And so along the x-axis here, this is common variants, and then over here is very rare. And you can see that very rare variants that have like a drastic effect result in what we would normally consider Mendelian diseases. And I'm sure in your nutrition studies that you have covered many of those.
We actually just talked about one this morning for those who are in class, PKU. So, yes.
And I hope it's also obvious to you that rare variants with small effect are very, very difficult to identify. There has to be usually a lot of what's called like a priori or prior knowledge about like why a region may be important due to its location, the variant's location in relation to an important gene ahead of time. Okay, so let's outline limitations and sources of bias. My hope is that you'll get out of today being able to outline some limitations and sources of bias in genome-wide association studies that impede clinical application. So what we've talked about up until this point is that associations have many possible causes. And the first is that we all sort of assume is that, oh, we're looking at association, this is as an association. One reason for that could be direct causation, that having a variant, let's just call it variant V, makes you more susceptible to disease X. Another possible reason for association could be what's called an epistatic effect. And people, this effect, you can't really distinguish in the results of your genome-wide association study, but it could be just that people with disease X are more likely to survive and have children if they also have variant V. And that's sort of a subtlety, but distinctly different from saying that this variant actually causes disease. The third one we've already talked about. Another possible reason for association is linkage disequilibrium. So the variant V marks an ancestral chromosome that carries another variant Y, causing susceptibility to disease X. So in this case, we say that V is associated with disease X, but Y is actually the one that's causing disease. Okay. Another possible reason for association is called population stratification, and this reason for association is due to that the population contains several genetically distinct subsets, and both disease X and variant V happen to be particularly frequent in one subset. So I'm going to show you an example of this because this can be a little unintuitive. Okay, so if this box contains our study population, we might incorrectly say that the blue variant is overrepresented in patients with disease X compared to controls. So in this picture, I think it's 13 versus 12 out of 20 for both groups. However, the association only occurs because there are so many more blue variants in population 1 compared to population 2. So there are 17 versus 9 out of 20 for both groups. And this is like, they're like, yeah, is that really a thing? And it is. It can screw up your association studies majorly. A very famous one, a group called published a paper. We're from San Francisco, and the researchers' names were Lander and Stork. And they conducted a genome-wide association study, and they showed that the HLA-A1 variant and being able to eat with chopsticks using a population from San Francisco, and clearly this variant does not confer the ability to eat with chopsticks. It's just that the people that the people with the A1 variant were more commonly Chinese than among white people, and so like you get this spurious association that's purely just a matter of the way that the population is stratified. So aside from self-reported ethnicity, can anyone think of any other genetically distinct subset that might cause you problems? So obviously you have different ethnicities could make it so that you can't really tell whether variants contribute to disease or are controls, but can anyone think of any other genetically distinct subset that could maybe cause you problems?
Sex.
Sex, yes, that would be important. You want to make sure that you have controlled for the number of males and females in both the disease and the control population. That's definitely true. Any others that maybe would be more problematic because we might not know it ahead of time. It's not necessarily easily reported. I'm going to just say it because whatever. So families, right? How genetically related you are. If there's a few cousins in your study population, you're going to have significant problems because they're going to have a bunch of variants in common just by the fact that they're related to one another, even if it's somewhat distally related. And if they also happen to have the disease, if they all happen to have the disease, it may not be because any of the variants that they share actually cause disease. It may just be because they happen to live in an area exposed to some environmental factor. And again, then their genetics wouldn't really be contributing to the development of your disease of interest. So if we know that we have a genetically diverse study population, makes it more difficult to identify susceptibility variants, why might this be a problem for translating findings from GWAS to the clinic? I mean, I think many of you spoke to this in one of the last questions from the vitamin D study. Why would it be like very? Why would these study populations confound what we can say about the variants when we're in the clinic and people say, oh, should I consider this variant or not? Basically just that they're only relevant to the population studied in the given basically what I'm trying to say is just that the study population that's used is really, the findings are only applicable for that study population. It may be that even though the minor allele that's associated with disease contribute is also is potentially also associated in a different population, but that always needs to be verified. There are numerous examples from the literature of variants that are associated with a disease, say in a white population, but in an Asian population or African American population, the association does not hold.
I'll just make a comment. I think you probably picked up on this from the vitamin D article, and they've talked about it a little bit, but unfortunately most people are getting a little better, but a lot of the genome-wide association studies are done in white populations, so it limits the generalizability, although I do think that's improving.
Yeah.
And I think funding agencies are looking really closely at the diversity of populations when they make decisions about research. So
yeah, it's really hard to balance, right? Like when you're a researcher and you're interested in getting at generalizability, getting at is this truly a real variant that's contributing to disease, and you make the assumption that this variant is going to be located near a gene that hopefully codes for a protein or is part of some regulatory system that has some cure effect. But to detect that effect, given the number of the unbiased nature of asking about the variants genome-wide, you have a really difficult time actually identifying anything if there isn't some homogeneity in your population. And so from like a very practical standpoint, people are like, oh, let's just make studies full of white people. And you're like, well, this doesn't actually apply to many other populations. And so funders, especially the NIH, they are there's like on all of your grant applications, you have to actually say like, you know, why you're specifically not including other ethnicities, and just saying that like it's hard is not really good enough. Like you have to actually be like show that, you know, especially if you're in an area that the diversity is present, or obviously if the disease is very uncommon in certain ethnicities, there'd be more leeway. So it's kind of always just this give and take of balancing like, you know, what is reasonable considering both like the need to actually discover true variants and the underlying biology, but also the generalizability and benefit for the taxpayers who are funding these studies. Okay, and then the last cause of associations that I want to talk about today is false positives. And I know you're all doing it's kind of boring, but I hope it's obvious to you that when testing a large number of variants for association with disease X, even without any true effects, some of the results will be called significant. And I'm going to just give a quick example to how extreme this can be, especially in genome-wide association studies. So let's say you're testing one variant for association with disease X, and you decide that 1 in 20 chance of getting a false positive is acceptable. And this is sort of where it's not exactly the same thing, but this in statistical lingo, we call that an alpha of 0.05. And this is also generally used in the biological literature as an acceptable standard. If you, at the outset of your study, you say, most people say that we have a 1 in 20 chance of getting a false positive, and we live with that. It's the price of doing science. Okay, now let's say that you want to test 100 variants. And again, you decide that a 1 in 20 chance of getting a false positive for each variant is acceptable. Again, your alpha set at 0.05. The probability of getting at least one false positive, I hope it's obvious to you that 1 minus the probability is equal to 1 minus the probability of no false positives, just because probability is always summed to 1. And in this case, the probability of no false positives is 0.95 times the probability of not one false positive, if we were only doing one test, would be 0.95, but the probability of having two tests that are not false positives would be 0.95 times 0.95, and again for all 100 tests. So the probability of getting at least one false positive in our 100 tests that we're doing is over 99%. There is pretty much an absolute guarantee that if you test 100 variants for an association, you're pretty much going to get at least one that is a false positive. And this probability of at least one false positive in a set of tests is called the family-wise error rate. And it's a huge problem, right? Because like if you you're definitely going to get a false positive, like it's going to be a waste of time and money, right? To be following up on these things that are just not true. But remember the goal here. How can we identify which genes are the most important in most patients? We don't want to spend a lot of money following up on variants that are false positives. We're calling all of these significant, and we need to really spend our time here and not here. Okay, so what do we do to deal with this? So this is what's called a multiple testing correction, and I'm just going to go through one of the more conservative and I think hopefully more intuitive methods for doing this multiple testing correction and controlling that family-wise error rate, that probability of getting at least one false positive. So the way we do that is we partition the total number of acceptable false positives among each variant tested, right? So if we say there's a 1 in 20 chance of false positives, and we do 100 tests, we just take 0.05 divided by the 100, and we get 5 times 10 to the -4th, which is a 1 in 2,000 chance of a false positive. So if we were doing 100 tests, we would say we need to have a 5 times 10 to the -4th value, a 1 in 2,000 chance of a false positive. It has to be past that significance level in order to say, oh yeah, this is meaningful, this is something we should follow up with when we move on to our next set of tests. And you can imagine this was just with 100 tests. Normally in genome-wide association studies, we're doing 1 million tests. And so this is where this 5 times 10 to the -8th comes from. And it can be adjusted slightly depending on how many variants you're actually interrogating in your study. That's what a really rigorous study group would do. And so you're familiar with the term. This is called a Bonferroni correction, and there are it's beyond the scope of this lecture, but there are less conservative methods that have been developed because obviously there's association between variants that are closer to each other, and so we can sort of lower the threshold and say, okay, it's acceptable to have a few more false positives, so we have more variants to follow up on. Okay. And then I want to talk about study design. So the two study designs that are the most common are case control, which we've already talked about today, just with the disease X versus control. And in a case control study design, you would have what's called a dichotomous character. Either individuals have or do not have the trait. and this tests for susceptibility genes. Another type of study design is quantitative. And this measures quantitative or continuous characters, and they test for what's called quantitative trait loci, which is just saying quantitative trait and like where in the genome is the association occurring. Okay, so the first study that I want to talk to you about was conducted by the Wellcome Trust Case Control Consortium. And this is actually a little bit old now, it's back from 2007. But in this study population, they used 17,000 individuals from the UK. And this was very early on before there was as much hullabaloo as to make sure that results were generalizable to other ethnicities. So everyone in this population was actually self-identified as white European ancestry. And the really cool thing about this study was that they had 2,000 individuals for each of actually 7 major diseases, and then they had 3,000 shared controls between for comparison with those 7 diseases. And so there were some more like fancy stats that was involved in just because there's some overlap between the control pools for each of these diseases. But it was still it was definitely one of the most like early groundbreaking studies when it came to genome-wide association studies. And they tested about 500,000 SNPs, and in the results they found that 24 independent SNPs, one of them was associated with coronary artery disease. I know one of the groups was looking at heart disease from the Design Your Own GWAS part of this lecture, so this has been done. And 3 were found in type 2 diabetes, among many of the others. I think they also did bipolar disease, and I know they did hypertension, and in many of those diseases, there were very few findings. I put this up here, this chart, this figure, because it's a very common figure that shows up in GWAS studies. It's called the Manhattan plot, because the idea is that if we find something, it will be like the skyline, and it'll be like a really high building. But essentially, along the bottom are the positions along the chromosome, and then the y-axis is the p-value, indicating whether or not variants at these specific positions are actually significant. So you can see there was the one significant SNP for coronary artery disease was on chromosome 9, and then there were 3 for type 2 diabetes, which I believe are here, here, and here. I'm not sure about these ones. This, and I think one of the, at least, I don't know if you guys have covered this yet. I know we did it and when I took this course, one of the variants identified in type 2 diabetes was FTO, the fat mass and obesity related gene. So this was one of the earlier identifications of that gene and its relation to obesity and obesity outcomes. And then some of the other genes are maybe less obvious. The other 2 from type 2 diabetes were Wnt signaling pathway gene, TCF7L2, and then the third was CDKAL1, and actually that has subsequently been investigated further and implicated in normal beta cell function. So this is sort of like one of the beginning studies that was important in laying the groundwork and really validating that this sort of approach could give you some interesting variants to follow up on in terms of pathophysiology. Okay. The second one I'm going to talk to you about is a genome-wide association study of macronutrient intake. So this study population was published in Human Molecular Genetics back in 2013, and it included 33,000 individuals from the US. This was, as opposed to the previous, was a quantitative study design, and they looked at the percentage of total energy intake from carbohydrate, protein, and fat which were calculated from food frequency questionnaires. And in this study, they actually tested 2.6 million SNPs. And they identified one independent SNP for carbohydrate intake and one sub-genome-wide significant SNP for protein intake that actually replicated in a second cohort. So it was not quite significant on the first round, but they replicated it, and as part of the joint analysis, felt that there was enough evidence to support this variant as potentially a variant to follow up on. The FGF21 cytokine, again, has been, which is this variant that was associated with protein intake, has since further studies have been done on it and is now referred to as the sweet tooth, linked to the sweet tooth phenotype, which is very interesting and random. Okay, and then lastly, I'm just going to talk to you quickly about the study that I'm at the beginning stages of working on for my part of my dissertation work. So I'm not running my own GWAS, that is like beyond the scope of a graduate student, but since there's been so many years have passed since the beginning of genome-wide association studies, there's a giant cohort out of Britain and the UK that has, it's called the UK Biobank, and the UK has a much more integrated health system, and they have designed this study looking at approximately 500,000 individuals from different regions of the UK, and they've genotyped all of these people, and they have considerable phenotype data on all of them, or many of them, and including, surveys on diet, obviously, which is what I will be using, but also mental health, their various health exposures, etc. And so it's sort of this very robust, publicly available resources for researchers who have specific questions regarding the genetic etiology of complex traits to design and submit for access to this resource. So that's what I'm in the process of doing. In terms of my study design, I'm hoping to look at the genetics underlying people who have a higher intake of fat and are drawn to high sugar intake, and I want to conduct this as a quantitative study and I'll be getting this data from 4 serial 24-hour recalls of people who just describing what they ate in the last 24 hours, and then there's a way to convert a prediction as to how much of their diet was primarily from total sugars and from fat. I'll most likely be testing around 800,000 SNPs out right now. I'm considering sort of the best way to do that part of the analysis. And then I'm also interested in a secondary aim, which is sort of outside of the scope of outside the scope of specifically the nutrition paradigm, which is looking at the genetic correlation with addiction phenotypes. And so I'm interested in sort of this idea that genetic variants could put you both at risk for these very highly palatable diets and also other addiction phenotypes. And those data are present in this resource through just self-report. So people answer surveys and they say, have you ever had any sort of problems with addiction in the past, what type, etc., and figuring out how you're going to quantify that in a rigorous way is part of what I will do as my work. Do we come up with some sort of score that they, had multiple addictions, or do we just say there's some history and we'll call that, good enough? So yeah. Anyone have any questions about this or anything really? That sort of brings me to the end of my talk. And here's these are many of the references that I used for this presentation, and feel free to come and ask me any questions that you have.
I have a question about your study.
Okay.
So what are you thinking about in terms of confounders?
So many.
Confounders.
So if I just talking about the like fat and total sugars portion of the study. As I mentioned, part of the reason that I wanted to pursue this was that this study of dietary macronutrient intake I was really interested in just being able to replicate these findings, right? This was 33,000 individuals, and the number of people in the UK Biobank that have finished the nutrition survey, the 24-hour recall, is 200,000. So the capacity to actually re-identify or validate the findings from this study is also really important, right? Like, is this really true? Like, do we want? I mean, FTO I think is pretty well validated, but there was other variants from this paper that were like somewhat significant, and whether or not we could actually validate that in a separate cohort. I also, in this study, the confounders that they considered were, they basically did the test for association, both including BMI versus not, in their model, which I think that I will probably include it because one of my main aims is to look at this genetic correlation with another phenotype, and so like I think that I'm not really interested in do not want like whether or not someone has a high BMI to confound like my findings. And then other things for obviously sex, age, and ethnicity will be all things that could be possible confounders. And there's like some of the challenges with this, especially the setup, is right that like being able to truly test whether or not the like environmental or patterning from childhood is really at the cause of why people prefer higher intake of sugar or fat is uncertain, right? Like you can use other stand-ins, like I've considered, do people have access to food, and what is a proxy that I could use for access to foods that are not high in sugar or fat, right? And you could consider like, I don't know, does anyone have an idea of what they might use that would maybe be a proxy for whether or not someone had access to foods that were not all high in sugar or fat?
Vegetables.
Yeah, like if that's part of their 24-hour recall, yes. Like perhaps their socioeconomic class, that would maybe give you like their family income. That would be something that you'd be like, well, like it could be just because like everybody in your case group, everybody who has a high fat or high sugar diet, like maybe they just like don't have access to other foods, and so that's why their intake is very high. It has nothing to do really with their genetics. It's completely to do with their environmental surroundings. So these are like this is sort of the challenge that researchers have to think of when they're designing their studies. Like, is there some other like easily accessible variable that could potentially help us, understand, okay, what is actually genetic and what's actually just some sort of environmental reason for having this trait or having this disease.
That's fascinating. Can you guys think of any other factors that might explain?
Yeah, I mean, like this could be really like you guys have spent a lot more time doing this, so I'd love to hear your thoughts.
It'd be interesting if you could get like their parents' recalls back too.
Yeah,
to kind of see if
that would be amazing. Like I mentioned, I sadly do not have the current resources to recontact a lot of these people, but actually the the UK Biobank, and for UK researchers who are well funded, you can request that if you use this this resource, that you could potentially recontact people. So if you had like the capacity to say like, oh, could you please let us know, like, you know, do you remember whether you ate, you know, you could give them sort of when you were a kid, do you remember eating vegetables 3 times a week, and come up with some proxies and some data for like, okay, like, do I think that this person really had some sort of like, you know, maybe their dad or their mom took them out every night and like they ate hamburgers. Like that could be something that you could assess for. But oftentimes we are limited just by the size and the capacity to do sort of these recontacting. And that also introduces bias, right? Maybe some people just remember their childhood better, and who's more likely to remember their childhood versus not, and does that have any, I mean, especially in looking at the genetics of addiction, you could imagine, well, that could be problematic, right? Like, it could be that, you know, many people who suffer from addiction, it's been demonstrated, have like a history of adverse childhood events, and so like whether or not they recall whether their parent did or did not give them to feed them the healthy food is going to be sort of all mixed up with that. So yeah, but yeah, that's awesome. Thanks. Anyone else have any? Do you have your hands on that?
I was just going to ask if you had any information about trauma at all.
Yes, they do. There is some. There is. They ask. There's so much data in this resource that I'm still like going through a lot of the variables. But yeah, they ask something about like, you know, do you remember if like you're, you know, were you home every day or something like that? Like they have a series of questions that takes them through like, did you have like, do you remember like not sleeping for a week? Stuff like they ask about anxiety. The timing is sort of always an issue, right? Like whether or not like some of the questions like are asking about like childhood, but some of them are sort of conflated with like, you know, they're asking about today, and is that really representative of when you were a kid? It's, you know, unknown or uncertain, unless you had like really good evidence from the literature that someone has demonstrated that this question is really well represented, really represents some, you know, proxy from childhood. Yeah. They do ask about like some birth exposures, which could be like if your mother was diagnosed with intrauterine growth restriction, which you can imagine might be important for obesity outcomes and diet patterning. But there's very little literature on that, so I'm sort of uncertain at this point. Anyone else?
Sort of thinking geographical, sort of like divide them regions-wise in some kind of way and see like what's the radius for you to get to a supermarket?
So they actually do have that information. They have both like a rounded and a raw value for latitude and longitude of each of the participants. And actually the exact latitude and longitude is like, you have to like special request it, because obviously that's like very identifiable. But yeah, that would be something that would be super interesting, right? Especially to tease apart this like genetic versus environmental components. If you could actually place some sort of score, like, okay, I know that I've mapped sort of where all the grocery stores are in the UK, and I want to say like, I want to give a score to how far each of these people, their latitude and longitude is from one of those grocery stores in terms of like miles or meters, I guess it would be, because it's the UK. But yeah, that's like totally something that I could consider including. Anyone else? Awesome. You guys have been great. Thank you so much for participating. This was really helpful for me, so hopefully you learned something, and I think you have the slides, so hopefully if you read a genome-wide association study paper in the future, you feel like you can actually like assess whether or not it was fitting to your patients and just of interest in general.